Did you know that cancer is responsible for more than half of the death worldwide? According to the World Health Organization, 53%, which is 9.7 million people died around the globe because of cancer in 2022. The consequences of getting the disease include but are not limited to the overall well-being of the patient, as well as the financial and emotional impact on their family members. While cancer is a complex issue and disease with many causes, molecular factor is one of them.
Among various molecular factors, the colony-stimulating factor 1 receptor CSF1R play a critical role in regulating the cell survival, the migrations, differentiations, and the cancer progressions. The abnormal activations of CSF1R has been linked to breast, lung, pancreatic, and hematological cancer, making it an attractive therapeutic target. The CSF1R inhibitors such as pexidartinib have shown clinical promises, but the resistance remains a major challenge, highlighting the needs of the novel CSF1R inhibitors. The cheminformatics and machine learning approaches, especially the QSAM modeling, offers a powerful tools to accelerate the novel CSF1R drug discoveries. In these studies, we apply the machine learning-based cheminformatics on the Campbell datasets to design and predict the potential inhibitors of CSF1R. The most promising candidates were evaluated through the biological testings and molecular docking to investigate their binding interactions and anti-cancer potential.
We began our research by collecting datasets of CSF1R inhibitors from Campbell database, specifically under the target ID Campbell 1844. The smart structures were then converted into 1D molecular descriptors using RDKit, including molecular weight, rotatable bonds, TPSA, and Morgan fingerprints.
As we can see from here are the raw data of the potential inhibitor of CSF1R that we have collected from the Campbell database. And here's the final data where we convert the mouse cells into the molecular descriptors using the RDKit module.
The prepared datasets were then used to train multiple machine learning models, including random forest regression and FNN. Hyperparameter tuning was performed using grid search CV to optimize the models, and K4 cross-validation was used with R² as the performance metric. The SMOT technique is used to increase the dataset sizes. From this, Random Forest emerged as the best performing model, which we later applied to screen the entire Campbell database for potential CSF1R inhibitors.
As you can see from here is a process of our random forest regression model training. And this is the hyperparameter tuning, which uses the hyperparameter tuning to find the best hyperparameters for our AI model training.
The Random Forest regression model achieved an R² score of 0.8935 on the test set and an RSME of 1908.14, reflecting a relatively small prediction error and confirming the model's robustness in capturing complex molecular relationships. The sharp XAI analysis revealed that molecular weight is the most influential feature in the model's prediction, followed by TPSA and Morgan fingerprint 1738.
The trained random forest regression models is deployed using a Flask-based web applications, and this is used to scan the entire 1.2 million of the drug-like compound in the Campbell database. As you can see here, this is the prediction results of our AI model. This is the compound name, while this is the predicted IC50. So the compound with the lowest predicted IC50 will proceed to the molecular docking simulations.
Top candidate compound identified by the ML models are then docked onto the binding site of the CSF1R protein with a search box using AutoDock VINA and the binding affinities. And the binding affinities are then obtained, and the molecular docking results indicate that our discovered drug achieves a binding affinity of negative 10.3 kcal per mole, which is better than the currently existing drugs. Finally, we evaluated the toxicity of our discovered compound using in silico prediction through the ProTox2 platform. The results indicate a predicted lethal dose 50 value of 2,500 mg per kilogram with a predicted toxicity class of 5.
Our project goes beyond computational drug discovery. It also aligns with several United Nations Sustainable Development Goals, such as Goal 3, Good Health and Well-being, Goal 9, Industry, Innovation and Infrastructure, Goal 13, Climate Change, and lastly, Goal 17, Partnership for the Goals. In terms of environmental contributions, our workflow minimizes laboratory waste by relying on simulations instead of repeated physical experiments. It also reduces the dependence on animal testing, making the process more ethical while simultaneously lowering the environmental footprint of research. Scientifically, our project accelerates the pace of drug development by quickly narrowing down promising candidates. It enhances precision in compound selection, ensuring that only the most viable molecules move forward. Ultimately, it contributes to the broader field of drug discovery, helping society move towards faster and more reliable solutions for human health.
Next, we are keen to be part of distribution of tasks in Thank you.